formal introduction, and then we'll revert back to that conversation, because I was thinking I'm recording and then people will be really cross because we missed the really good bits. So I am delighted to introduce Dr. Emma Harvey. We work together on the Connecting STEM Teachers programme with the Royal Academy of Engineering, and I'm just in awe of everything she's done. So from science to education to working for the Academy with me, to being a data analyst, to then co-founding the 5 Hour Club, which if you haven't seen it on LinkedIn, Instagram, there's a podcast, then seriously, you need to follow it because it is incredible. So we were just waiting for a couple more people to join, and I was just about to say, and I thought this is something we need to share, that when I went to do my degree in marine biology, I really did think that all I'd be studying was marine ecosystems. And that really isn't the case. When I went to study at Portsmouth, we were given a real variety and we studied different ecosystems. We had a real wealth of different areas of biology that we researched, that we looked at. And so I think, when you're going into your university courses, have that open mind that actually you end up studying a variety of different disciplines. And that's really valuable because actually you might change your direction. So I'm going to hand over to Emma, who's going to give us a little bit of a journey into her research, and I'm absolutely buzzing for this. So over to you, Emma.
Okay, thanks so much, Laura. Okay, so I am not going to get this started by showing you my name or showing you my thesis title. I am going to get this started by asking you 3 questions. So if you're watching the recording, I just want you to have a think about this, right? And do you actually know anything about this topic whatsoever? Now, the image might give away a little bit that we are going to be talking a lot about the Ice Age. So first question in your mind, do you know when the last Ice Age was? And what do you know about the Ice Age? The second question is, where did the plants and the animals go? So if we had an Ice Age that looked something like in the cartoon, where did a lot of the ecosystems that we have today in Europe in particular, well, where did the plants and animals go? And then finally, how do we actually know what happened if the Ice Age happened however many years ago? How do we actually know this? Now, these are the 3 key questions really that I'm going to be talking about today. But as I say, have a think to yourself, do you know any of the answers to that? Now, I've given this a title of A Tiny Plant's Big Secret and how we can unlock Ice Age mysteries using genetics. So I've kind of given away one of the answers to the questions. How do we actually know what has happened in the past? Before I give you away kind of some of the answers to the questions that I got you to think about, let's have a look about how I am going to jam pack this session today. Now, apologies in advance if you hear slight shouting. That is my children causing chaos upstairs, but hopefully there won't be too loud. So I'm going to try and cram 4.5 years of work into 50 minutes, and I'll try my very best. And I want to take you on a journey, not just on kind of what I did and the analysis and what we found out, but actually on a practical level. What's it like to do a PhD? What's it like to do genetics research or on this sort of research? So the first thing I'm going to talk about is a bit of the science behind the background and the theory behind my PhD. I'm going to talk about what we do. Now, I use a lot of we in this because I did have a lab partner who did a lot of the work with me and my supervisors. It was a team effort. I'm going to talk about the analysis, so how did we actually kind of find out these mysteries, and what were these mysteries? What did we find out in the end? And then what happened next? So Laura, in very kind words, mentioned a little bit about my journey, and I thought I'd relate kind of, in hindsight, what I tell my younger self about all the skills that I gained studying science in my degree and in my PhD, and how they apply now. Okay, so the fancy pants title for my PhD is a phylogeographic study of Arenaria ciliata and Arenaria norvegica in Ireland and Europe. Really, it's a tiny plant's big secret. So the first question is, when was the last Ice Age? I'm going to show you a graph. We're not going to look in too much detail at this graph, but on the bottom you can see however many years ago, and on the y-axis over here you can see the temperature. Now, the key thing I want you to take away is this is a long time ago. We're talking, there have been cycles of Ice Ages really for 2.5 million years. So this is even just a small segment of that. Now, if we're talking about the very last Ice Age, The second thing I want to talk about is we are currently in present 2025 in what's called an interglacial. In other words, we're not in an ice age. So we are above this line, we're above 30 degrees Celsius. Now, because of global warming, actually, we are overdue an ice age, but because of global warming, we are not in that right now. That's a topic for another session. Now, if we have a look down, so the last glacial maximum, i.e. the last ice age, was about 40,000 years ago. And I'm going to show you a picture and try to imagine what that would have been like. And the ice sheets would have melted about 10,000 years ago. So that would have been coming into the interglacial, kind of a little bit what it is like today. Okay, so I've got a map of Europe. What you might recognize some bits, but it looks a little bit different. Now, the first thing I want you to imagine is we need to get an understanding of like 1 kilometer, right? So imagine your local coffee shop, or think about somewhere that's 1,000 meters away, 1,000 really long steps away. So it's about, what, 10-minute walk. So imagine yourself walking for 1 kilometer. That's about how thick the ice sheet would have covered Europe. So about 1 kilometer thick, this really thick ice sheet, and that's what that white area represents. So this ice sheet would have covered all of Ireland, the majority of the UK. And there's a couple of things to take away from this. So if you've got this really thick ice sheet, number one, the plants and animals have to go somewhere. A lot of these plants and animals are more Mediterranean, they're used to warm climate, and simply they cannot live on this thick ice sheet. Number two, you might also, one of the reasons why it looks a little bit different is you've got these areas of land that are protruding above the sea. Because if you've got so much water trapped in that kilometer thick ice sheet, the sea levels would have been much lower. So they would have exposed areas of land that we call land bridges, and that would have enabled plants and animals to move across Europe during this time. Okay, so again, give a picture, and that's what it would have looked like. So the vast majority of Northern Europe covered by this ice sheet. So the second question is, where did the plants and animals go? Because we know there would have been some extinctions, but for the vast majority, they did survive. So we've got 3 hypotheses. So when I started this PhD, there were kind of 3 really accepted hypotheses. The main hypothesis was that there was an Irish-English land bridge. Ultimately, the plants and animals from Ireland in particular and England, they would have basically gone down to southern Europe, like the Pyrenees and the Alps, for a little holiday for many thousands of years. All right, and that's in areas what we call refugia. So these are areas that would have contained all of these plants and animals, and they would have stayed there for that time. Now, if you can imagine that ice sheet retreating, moving away, it's melting, then what would have happened is the plants and animals would have migrated, come back from their little holiday, up north across from France to England, or across a land bridge from Spain to Ireland, which is the second hypothesis. So actually there are relatively shallow seas across from Spain to Ireland. And that is another hypothesis that they would have migrated across northward. Now this is, in the grand scheme of things, relatively recent, is 10,000 years ago. So we're talking about, when we're talking about plants and animals and evolution, 10,000 years sounds like a lot. It's actually really nothing. So this is the kind of the time that we're talking about. And then finally, there was, at the time I started my PhD, a final hypothesis that some people suggested, but it wasn't widely accepted. And that is, some of these species survived in these refugia, but actually in or around Ireland. And I'm going to tell you in a moment why some people thought they actually survived in Ireland. So last question I got you to think about it. Well, how do we know on Earth what happened? Like, this is such a vast, there were little animals and squirrels like taking notes. Well, one day we went to the south for a holiday. So obviously, how do we know? So number one, so you may know in terms of, so one of the reasons why I loved and I chose my PhD, in its very nature, PhDs are very specific and you become an expert in that particular topic. But my particular PhD actually was really multidisciplinary. So in order to understand what happened, I needed to kind of understand, well, what was happening climatically at this time? What was the environment looked like? Now, I didn't look at ice cores, but I certainly looked at maps to try and help me identify and understand, and even time scales. So ice cores, so we know kind of approximately what the climate was like and how cold it was because of these ice cores. We also know something called peat cores. So similar, you get a core, you put it in the bottom of the lake or a bog, and you can actually get some samples of pollen. So you can actually tell from, for example, 5,000-10,000 years ago, what the ecosystems would have looked like at that time. So would it have been a tundra environment? Would it have been milder? And actually, you can also tell the climate because of that. Now, I appreciate, and I probably will say throughout this lecture, pretty much every topic I mention, you could do an hour-long lecture on it. So this is very, very shallow, just showing you there are different ways you can get evidence, because the key evidence that I used was genetics. Now, what I want to say, so this is my absolute bread and butter, and this was the tool I used to understand where plants and animals, were these particular little plant species, and what happened to them. Now, I just wanted to show you this map. Now, this is actually human migration, in particular in the UK, in a more relatively recent timescale, so about 1,000 years ago. But I just wanted to show you that what we did as a tool, you can do with any living species, including humans. And I love this map. I just find it so interesting because ultimately, my PhD was population genetics. So what we can do is imagine a population. So you can have a population. A population really is just a group of individuals from the same species in a location. So for example, if we look at humans, we can classify Cornwall essentially as a population of humans. And what they did was you can get DNA samples from living humans, and they did this. This is a study in the journal Nature, and they went around the UK. They got loads of analysis from DNA, and what you can do in a population is you can essentially get a genetic fingerprint for that population, and you can tell where did those humans come from, where, so are they Anglo-Saxons, are they Vikings, are they, at what point did you have that wave of immigration, or are they more isolated? And you can see from the colors represent the essentially that genetic fingerprint of the population. Now that red one is quite interesting. So they tried to understand also, was is there like a distinct genetic pattern for the Celts? And actually there isn't necessarily. So that red kind of pattern, it's what we call homogeneous. It's kind of spread out. There's no distinct kind of pattern there. But you might be able to see down in Cornwall is pink. Yes, there is a genetic, a distinct genetic pattern for Cornwall. The same for Devon, a distinct genetic pattern. And the same for North Wales and South Wales. And why is that? Well, generally, again, population genetics, you could do a whole module on this. Generally speaking, if you are isolating those individuals and they are inbreeding with each other, you don't have to infer anything about people from Cornwall. this is just population genetics. But what is going to happen is that they are going to have a distinct genetic, let's say, fingerprint for that population, whereas everyone else in the UK are breeding with each other, and actually I don't have that distinct genetic population. So actually what I did was fairly similar. And one of the reasons why, so we looked at this small little Arctic alpine plant. So what does that mean? Well, it exists in the Arctic, so very cold regions, or in alpine regions at the very, very top of the mountains where it's very cold and icy. So we looked at this little Arctic alpine plant, number one, because if anything was going to survive the ice age, it was going to be an Arctic alpine plant. And number two, this was what we call the Irish puzzle. Now, do you remember I said that kind of hypothesis of the plants and the animals, and they migrate northward from France across to England and then to Ireland? So down in the south of Europe, it's much more biodiverse. So there's about over 3,000 species of flowering plants down kind of in the south of Europe. In England, it is more than Ireland. So it's about 1,500 flowering plants. And in Ireland, it's about 800, 850 flowering plants. So that kind of makes sense if we're talking about the migration. So you would have less biodiversity as they migrate northward. But there are 15 plants that occur in Ireland that don't occur in England. So that doesn't really fit with the hypothesis of that northern migration. And this is one of those plants. Now, on that map where you can see its little red dot and it says time 4, that is the only location it occurs in Ireland. So so that little red dot occurs there, and it occurs in the south of Europe. Now, that blue dot I'm going to talk about in a moment. Okay, so that's the Irish puzzle. That's one of those anomalies. So there's 2 reasons why we looked at that little plant. So this is the only area that it exists in Ireland. It's a place called Benbulben in Sligo. It's this incredible landscape. It's essentially a mountain, and with a flat top, and that's where you find that little plant. Now, I also looked at a plant called Arenaria norvegica. Very similar. If you were to look at the 2 plants, you might not be able to tell the difference. This is a really interesting one, and you only find it in one location in Ireland, with the little blue dot called the burn. Now, the burn is a really magnificent environment. If you haven't been there, you have to go. It essentially looks like the moon. So it's what's called a karst carboniferous landscape. Essentially, the glaciers kind of removed a lot of the soil, and it's this really unique ecosystem in Ireland where you get Arctic alpine plants living side by side Mediterranean plants. It's got a really interesting microclimate as well. And here's just a picture of that little plant in Iceland. So you can imagine the kind of environment that it's living in nowadays. Okay, so I'm still on the background. Now, I also want you to try things. So we're going to get into the nitty-gritty of the genetics, okay, without going into too much detail. So what I also want you to appreciate is thinking about, well, I was looking for DNA in plant cells, but where do we find DNA in plant cells? Well, there are 3 places. So we've got obviously in the nucleus, so we've got the nuclear DNA, and we also have DNA in the chloroplast and the mitochondria. Now, the really interesting thing is, so I looked at DNA from the nucleus, and I'm going to very briefly talk about one method, and I also looked at chloroplast DNA. Now, something that's really interesting about chloroplast DNA is that it's only inherited from the mother. Now, the thing about the chloroplast DNA, and the reason why we looked at different types of DNA, is they give different, I suppose, pieces of history because of the way that they evolve. And I will talk a little bit more about that later, the more that we go into it. Now, again, I don't want to go into too much detail, but this just gives you an example of, let's say, the DNA that's in a plant called Arabidopsis thaliana. It's a pretty standard model plant that we use in genetics. And what I'd like to show you is that I looked at particular genes not from this plant, but this just shows you kind of a picture that there are many, 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 many genes in the chloroplast DNA. So in this particular case, it says 154,312 base pairs, but what did base pairs actually mean? Now, I'm sure you've come across base pairs, but in order to enter my brain and my analysis, I kind of want to show you where I'm at. So when we look at these genes, essentially these sections of DNA, when I talk about a base pair, I am in one sense talking about that base pair of the DNA, of the A and the T and the G and the C. But really what I'm talking about is a line of letters, because fundamentally scientists are lazy and we like to be efficient. And when I say base pairs, let's imagine this is a tiny gene, okay? I'm going to count this as 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. That's 15 base pairs long. But wait just a minute, I'm not showing the corresponding strand of DNA. I'm not showing, for example, the C and the G and the T and the A. Well, scientists are lazy. We know already that the C goes with the G and the T goes with the A, so we don't need to. So just to let you know, when I show you that series of letters, that genes, that's what we're interested in. Okay, so that gives you a little bit of the background. Now I'm going to talk about, well, what did we actually do? Okay, so in order to understand this kind of, and investigate these three hypotheses, we focused on the two. So technically, Arenaria ciliata is an Arctic alpine plant, and Arenaria norvegica is an Arctic plant. Now, so the plan was essentially we needed to hike all around Europe. We had to figure out how to climb a mountain, how to find the plant, and how to go to Svalbard and Iceland. So a lot of it was a lot of problem solving, because I'm going to be honest, I didn't climb a lot of mountains before I started my PhD. So how on earth do you do this? So what I did was I made a lot of friends, and I did a lot of planning. So in my first year, I was thinking, okay, well, here's a problem. Let's figure out how to climb a mountain, how to sample these plants. Now, I needed to go around Ireland, and I was actually sampling other plants in addition to this as well that was used for other research. So ultimately, going around Ireland and going around Scotland and England wasn't that challenging. I can speak English, I can figure out, where to go. I can learn how to read a map and do all that. But how on earth do I navigate the Pyrenees and the Alps? So what I did was, so when I was planning this, I was very lucky and I got to go on a course on Salzburg, and that's where I made some friends. So I managed to join them on expeditions for 2 summers in a row, all around the Alps. So one time we went to the Austrian Alps, the other time we went to the Italian Alps. So essentially every summer for 3 summers in a row, it would just be going around climbing mountains. Or luckily enough, I had scientist friends in Svalbard, so they would send me their little samples. It would also be, as you can imagine, a lot of time then in the lab. So it literally, and I'll show you some pictures. So I would go to the top of the mountain, I would get the leaf, put it in an envelope, take it home, and then extract the DNA, which I'll talk about in a moment. Then the analysis. Now, I did a little bit of analysis throughout the years, but really it was in one big giant go at the end. And then finally the thesis write-up. So when you're writing a thesis that's like 250 pages long, if not 300, you can start to write up the theory, the introduction, but realistically, I locked myself in a room for 6 months, deleted Facebook, and right at the end, and I just sat down and I wrote it all together at the end. So yeah, so although it says year 1 to 4, it really was at the very end. Yeah, so this is what I looked like climbing. I just wanted to get you an impression. So funnily enough, of all the places we went to in Europe with my lovely lab partner in Shandong, the most challenging hikes were in Ireland and Scotland. I think particularly Scotland, because it was wet and rainy and oh my goodness, it was challenging. Whereas when you are in the that's my lovely gang from the University of Salzburg, when you're with Austrians who know where they're going, it is much easier. But also they have like really clear paths, and the weather is much nicer. So ultimately what we do is we'd get an OS map, and so this was Ben Bulben, where I said in Ireland, where the population, that one location of where Arenaria ciliata occurs. And essentially what I do is I would just map out, okay, what I would literally just have to go to the top of the mountain and walk around and look for these plants and map out where am I going to define what a population is, and how to make sure I can get a really good sample size of this population. So I decided that there were 4 populations in total, because actually it was quite a large area. And this just gives you an impression also of kind of like the size of the populations, and just again, just the landscape. It was incredible. After a while, like, I mean, it was quite fun. You can imagine you would start to identify, okay, this is exactly the sort of area that it would grow in. Oh, that looks like a plant. Oh yes, that's it. And it's just a tiny little, it really was like a giant treasure hunt looking back on it. And if you'd like to know if I got lost, yes, I got lost twice. Because honestly, there were some areas that looked very, very similar. Now, I think I'm playing myself down here. I did end up becoming quite a good hiker in the end, because you learn very quickly how to do that. And again, here's just some to show you like where the populations were. So all located throughout the Alps. And we also drove in Spain. So we went to the Pyrenees and we went to a place called Picos de Europa. And this was much nicer because I went with my PhD supervisor who booked some hotels as opposed to camping in remote locations and hostels. But it was all fun. So what we do is, with these tiny little plants, we get the leaves, put them in an envelope with some silica gel. You might find those packets in the bottom of your handbag. And what it does is removes the damp, make sure they're nice and dry. And then back in the lab, I would spend the winters doing thousands and thousands of DNA extractions. Now, like I said before, you could literally spend an hour talking about a DNA extraction, and I won't, but ultimately, so back in the lab, doing the wet lab work was just a lot of experiments. And generally with genetics experiments, the majority of the time, it doesn't work. So it's a lot of problem solving. Okay, now back to the locations of the DNA in the plant cell. So the two key locations I looked at were in nuclear DNA, so from the nucleus, and the chloroplast DNA. Now, back to, again, this picture of what the chloroplast DNA looked like. So we identified a range of different genes that we looked at for this chloroplast analysis. And so what I just want to kind of highlight is, when I say a lot of the genetics, I kind of just want to give you an idea of what it looks like in a genetics wet lab on a day-to-day basis. So really, we found these were the genes that we looked at. But what it actually means is, we know other researchers found these particular primers. So essentially just a short segment of gene that will bind to the target DNA that we want. But just because we know the primer to order doesn't mean we know that that particular temperature that particular researcher found worked best for them, for their plant. It doesn't mean it's going to work for our plant. So we had to spend a long time doing something called PCRs, or PCR, polymerase chain reactions. Now, you may have studied it, you may have heard of it from the pandemic. The PCR brings it all back. It is it's such a widespread technique in genetics. So you can use it for paternity tests, you can use it in forensics. And essentially, I used it on a pretty daily basis to understand, did my experiment work? So really, imagine you are doing a bit of DIY in your house, right? You've got a room and you want to paint it. Well, you're not just going to start painting, you're going to prep the walls, and you're probably going to spend 90% doing the prep, taking off the wallpaper. Don't get me started on the wallpaper. Taking off the wallpaper, seeing what plaster is crumbling off underneath, prepping everything, right? That's essentially what it was like. So, I mean, really, it was, and I'd like to talk a little bit about transferable skills, because what you're doing in the lab, my goodness, you need to be resilient, because again, if it doesn't work, you're like, okay, I'm gonna have to try again. But let's say I want to determine, okay, I want to, and I think the analysis will hopefully make a bit more sense when we move on to the analysis. But let's say I'm like, okay, I want to look at this gene. I know the scientist has identified this sequence at this particular temperature. What I'm going to do is I'm going to plop these reagents all into, my little vial. I'm going to put them in a thermocycler. So essentially what we're doing is just like photocopying DNA to make sure that we've got loads of copies of it, that we have enough for analysis. And I'm going to try it at 56 degrees and see, and run it on a gel, which I'll show you in a minute. And if I get nice bright bands, I know that is optimal to then send it off for sequencing and get back results. It's not as easy as that, because this teaches you the scientific method. Okay, so let's say you get something back, it didn't work. I'm going to try again. So why didn't it work? Well, it might have been one of the reagents. And what you learn is process of elimination, but really the scientific method, in that I'm going to change one variable. I'm going to figure out, was it the reagent? Okay, we're going to remove that and try it again. We're going to remove that and try it again, and keep on doing this again and again and again. So a lot of the majority of the work was just in the lab, doing, you know, these experiments again and again and again to try and get these beautiful bands so that, you know, you can send it off for sequencing. So so as I say, spend a whole hour looking at electrophoresis, but just in a nutshell, it's just to give you the idea that on a day-to-day basis, you are doing these PCR reactions to identify the optimal conditions, just like baking a beautiful cake, the optimal conditions, and then you replicate that, and then finally you can send them off for sequencing. Now, that is just for the chloroplast DNA. I'm also going to talk very briefly about, do you remember I said about the DNA from the nucleus? Now, again, I am literally just going to very crudely mention the other technique that I used, and it's something called amplified fragment length polymorphisms. In a nutshell, I chopped up DNA using enzymes, and that's essentially what it is. So I chopped up, again, this DNA from the nucleus. Now, one of the reasons why the DNA from the nucleus differs from the chloroplast, if you can remember, I said from the chloroplast, you only inherit it from the mother. So actually, it's much more slowly evolving. Whereas the nucleus, you have DNA from the mother and the father. So you're going to have lots of more variation. You're going to have lots of more mutations. So you are potentially, so this is used a lot in population genetics, where you're trying to detect, imagine two little plants living side by side. How are we going to see any genetic variation? This is going to really help with that. Okay, so hopefully that's enough of the absolute bare bones to understand. Okay, so we've got these chloroplast genes. We did a lot of PCRs. We send them off for sequencing. Then what happens is, honestly, the really exciting stuff. So we've got the chloroplast DNA. We've got the AFLPs. They're coming back. And I'm going to show you what essentially they looked like. I mean, God bless my little MacBook. It's a little old now, but that did a lot of the heavy lifting of the analysis. So now I did have a sequencer in my lab that looked something like this, but there was a stage right at the end of my PhD where things weren't working at all, and it wasn't the reagents, it wasn't the temperature. I really narrowed it down, and it turned out our sequencer had broke. And I just couldn't believe it. So there was panic stations, because that's the thing about sometimes about doing a PhD. You're like, I'm not going to finish my PhD if I don't get the analysis. I've done all my wallpapering, my plastering, like I'm just about to get to the good stuff. So right at the end, we figured it out, thank goodness. And we were able to get our samples and send them off to, I think it was Sanger sequencing. And what they give you is an email with data back. And it's very exciting moments. And I'm just going to show you what it kind of looked like. So imagine for the chloroplast DNA, all right, and imagine you've got one little plant and the next little plant living side by side. And what I want to highlight, remember I said, you know, all I care about is the letters. I don't care about the two strands. Really, what is informative is this sequence of letters, right? And the reason why that is let's say you've got, let's say, plant A, and then underneath you've got plant B, and then underneath you've got plant C, and underneath you've got plant D. Really what we're doing is bioinformatics. Now, the great thing about bioinformatics is you can download DNA for free. So it's incredible, actually. But we did do the wet lab work, and this is just an example of what it looks like. Now, what we do, so then we align them. And the reason why we align them kind of one on top of the other is you can see the mutations. So for example, if anywhere you see a blue, what is that, rectangle, you've got a mutation. So you can see here, so you've got a C, then that's an A, and then underneath you've got a C and a C. So that's a point mutation that's changed to an A. Anywhere where you've got, you can have deletions where you've just got areas that are deleted. So for example, that red area. And even visually, you can start to realize, oh, hang on a minute. Actually, even on a population level, you can start to see individuals that are more closely related to than others. And this is exactly the same like the map that I showed you of the UK and the humans. This is what it looks like. You are just aligning those long sequences of DNA and looking for the similarities and the differences. But luckily enough, we have computers for that. Now the AFLP fragments, remember I said, so this is from the nucleus. We chop up the nucleus. Now, all I want you to look at is, we're not going to look too much at the tree to the left. If you even look at to the very right, we've got two distinct species, and this is just an example. So we've got C. coli and C. jejuni. If you just have a look at the, so this is what a PCR, the results of a PCR on electrophoresis would look like. You can even just visually see, oh, there's a pattern there, actually. So there's a pattern for that species on the bottom and on the top. So visually, you can see that. Again, we are looking for similarities, we're looking for differences, and that's what the computer does. Now, the AFLPs, this is what I looked at a lot of the time for many, many, many nights and days and mornings. And it essentially looked like this, okay? Now, what I want you to imagine is, well, what on earth is this? How do we convert this into useful information? And what does it all mean? Okay, so in a nutshell, the AFLPs, so remember I said they chop them up into little fragments, and then they're separated depending on their size. So, so really, these are just like chunks of DNA fragments. But that can give you a genotype of a population. That can give you similarities and differences between these plant species. So I'm going to do a little exercise. And let's imagine. So the first thing that you need to do is quality control. And what we're interested in are these peaks and comparing them. So like I said before, you align the different, you align the individuals. So one on top of the other so that you can directly compare them. So, I mean, again, the grunt of the work was trying to get it to work. The grunt of the work was doing quality control and making sure, oh, I'm not going to quite accept that because I don't think that's a good enough sample, and it would just look like a wavy line. But when you get the beautiful peaks that look like this, then you can decide, okay, so at the bottom, you can see the size of the AFLP fragment, so the base pairs. So that just tells you, for example, here, 140. Well, that's a fragment of DNA that's 140 base pairs long, and that's really it. But the really cool thing is that profile and comparing the profile. And I'm going to show you how the computer does this. And I kind of had to help the computer along really with the getting the data together. So let's imagine, right, what we're interested in are these three peaks. So let's say where that little arrow, where that little black arrow is, all the way to the left, up here, that top peak in the middle, and down here to the right. What we're going to do is convert it into binary. So imagine here for plant A, we have identified, yes, that peak occurs at that position. So I'm going to say 1, yes, it occurs. The next peak, yes, you've got a nice fine peak over there. We're going to call that a 1. Is there a peak where you've got that third arrow? No, there's no peak in this particular instance. So it's 110. The next case, yes, we've got 110, the same. In the third sample, we have 1 peak here, 1 peak here. So it's going to be 111. And the last one, we've got no peak there, no peak there. And it's going to be 001. So already on a simplistic level, hopefully you can start to appreciate, oh, we can clearly see there are similarities and there are differences. But back to the analysis. Once I had, so we did all the wet lab work, we did this analysis, but the majority of that analysis, and I do work in the data world now, the majority of data actually is getting the data and cleaning the data. This is the stuff that kept me up at night. And did I give my family a PowerPoint presentation about the results? You bet I did. It was so exciting. Very sad, but very true. Okay, so what did we actually find out? Now, I am, as you can imagine, I'm skipping a lot out about, again, the details of how we actually did it. But what we did do is, once I had those genes, that chloroplast genes lined up, I was able to do kind of the key analysis, which I'm going to show you now. Once I had that AFLP, those lovely profiles lined up, and I was able to do this great analysis, I'm going to now show you 5 images that will hopefully show you why I got so excited. And maybe it's not just me, I don't know. So I'm going to take you on a little journey that will hopefully explain the mysteries that we found in the end. Okay. I'm not going to spend too long on this one, but I am going to go back to the map of Europe, and I am going to start to use the word haplotype. Now, imagine a genotype. So a genotype is essentially the genes that occur in that individual or in that population. Now, the reason why I'm using the word haplotype is because we are looking at chloroplast DNA. And remember, I said chloroplast DNA, you inherit from the mother. So it's essentially half of that genotype. So just in case you see that where you're like, what on earth is going on? I'm just talking about the chloroplast DNA. Now, essentially, just like the UK human, we can give that genetic fingerprint or haplotype to a population. And we can start to understand a few things. So, for example, what was the genetic diversity? How closely related were the Irish populations, for example, to the Spanish populations or the populations all over the Alps? So that's what we did. Now, we started to find, and this is where it starts to get a little bit interesting, for a more recent colonization. So imagine plants and animal species went across that land bridge from England to Ireland, and for some reason became extinct in England. You would imagine very little genetic diversity in the last 10,000 years. So you would imagine a kind of, do you remember the map of the UK where you've got that red splattered all over it? Like, that's not very genetically diverse. You would imagine something like that. We were starting to find a few haplotypes for that single population in Ireland. So in that area called Ben Bulben. So that was really interesting. We also found, so I'm going to move on now onto the next picture. So that was the first thing that we found. Then we started to make what's called a phylogenetic tree. Now, in a nutshell, a phylogenetic tree is essentially like a family tree. And if I just talk you through some of the key things here, and again, why it's so exciting. Now, when I just talk about this phylogenetic tree, in particular for concatenated chloroplast haplotypes, concatenated, it just means we got all of those genes, all of those 5 genes, and we put them together. Because the more data that we have, the more reliable our evidence is. Okay, so to not talk about this in too much details, but let's have a look at those Ben Bulben populations. So if we have a look at group A, we're starting to see a little story emerge. So the BB, that's the Ben Bulben, right? That's the particular one in Ireland that we're interested in. Now, we know that if you are located on a phylogenetic tree beside each other, essentially you were more closely related to each other. So there are less mutations. So we know that this particular group are closely related. So we've got Picas Europa, Austria, we've got Ireland, and we've got Spain again. We also have another group, so we've got the Pyrenees and the Alps. So nothing too exciting. So you could potentially infer that maybe Ireland did actually come from this Austria, or it came from Spain. Now, let's have a look at group B. Now, group B, we know that this is kind of where it gets really weird. So we've got a group here from Spain And then in the middle, we've got that Araneri norvegica, that different species. And we've got 2 haplotypes, so 2 genetic fingerprints. We've got one from the Burren, that other location, Ireland and England and Scotland and Shetland. So that light blue is this over here. So that light blue, we've got 1 haplotype here for that tiny, tiny little population and in England. But we've got another one, we've got another haplotype occurring in that tiny little look that occurs in Iceland also. Okay, so that's kind of interesting that's got those 2 different haplotypes. But the really weird thing is that, wait just a minute, you've got Ben Bulbin again. So you've got 2 other haplotypes, 2 other genetic fingerprints, and it occurs with Araneri norvegica, which is a different species from rum. Okay, so rum is over here. Okay, so that's got a distinct haplotype, but it's very closely related. So we're like, hang on a minute, these are 2 different species. They are more closely related to the other individuals from the same population in Ireland. This is looking very exciting. This is looking more like it's matching the hypothesis that it survived the Ice Age, because we're looking at multiple haplotypes. We are looking at this really interesting rum association with the other species. And we're looking at there's a few other things that the phylogenetic tree can tell us. So the length of it can also indicate how different it is. So for example, if you have a look in the very bottom, so group C, you can see is genetically diverse. And because that's the West Alps, we would expect that. And in the very bottom, we've got a very interesting one. So it's very long. So ultimately, we can tell where they all originated from. So in the back before, in the long, long time ago, all of these would have originated from that one in the Western Alps. But we're talking hundreds of thousands of years. Okay, so we've got an interesting picture emerging here. And we know that Ireland is genetically diverse. We know we've got this kind of really weird association with the Araneri norvegica and rum. What's going on? Very briefly going to talk about this. Now, what the phylogenetic tree doesn't tell you is how many mutations occurred. Now, you can also do what's called a haplotype network. So you can have a look and you can actually literally count the number of mutations that have occurred. So in other words, have an A changed to C, how many times has that happened? And you can tell how genetically distinct it is. And very briefly, if I just have a look at haplotypes number 1 and 2, that's the Ben Bulbin, that's the one of interest. Okay, so they're right in the middle, and they're quite distinct from the other haplotypes. So they are actually unique haplotypes, and that's the key here. The other one, haplotype 20, now that has a box around it, which indicates that it was an origin, so a population of origin. So, and that is that really interesting one that's in the Burren, and beside the one that's from rum. So again, the more analysis we did, the more that it was confirming they are very distinct. They're quite relatively diverse. It's very, very interesting. Now, I'm going to talk about, so that was for the chloroplast DNA. Now, the AFLPs reassured us in a little way. So this is from the nucleus. And this can help us to make sure, well, are these 2 distinct species? Are they subspecies? But actually, what it did confirm to us are, yes, they are distinct populations. And yes, they are distinct species. So we've got the Ben Bulbin up in the very top. So that's in one group there. Then we've got Araneri norvegica, so in the middle, and then we've got the alpine populations down at the end. So Araneri norvegica, it did confirm, yes, it is its own distinct species. But what may have happened is so at this point we were thinking, okay, we do think that there was a refugia in Ireland, potentially off the coast of Ireland and in Rum, where Araneri norvegica and Araneri ciliata were co-occurring and reproducing with each other. So, and that's why we've got that pattern from the past. So by looking at different areas of the DNA, it actually tells us a different story, whether that's an evolutionary story or whether that's a population genetic story. And I'm just going to finally finish with this because it's a pièce de résistance, our final confirmation of, yes, this actually did survive the Ice Age in Ireland, and not just over 20,000 years. We're talking over 500,000 years. So this is called an evolutionary clock. Okay, so you can actually date when they evolved, essentially. So it looks like a phylogenetic tree like we saw before, but in the very bottom, you probably can't see it, it's quite tiny, but it says million years before present. So you can, so I did spend quite a few months trying to get this to work, but we got it to work in the end. And yeah, incredible results. And the key thing is, in red, you've got that Ben Bulbin population. Again, they are in two different clades, essentially. But what we can tell is if we go back to the last point in which they, what we call diverged, you can tell, so for this particular one, so the last point it diverged, if you follow it down, it was over 500,000 years ago. So actually did survive the Ice Age over 500,000 years ago. And yeah, we had kind of multiple lines of evidence from the genetics to show that. So what happened next? I'm not going to talk into too much detail about this because I do want to leave some room for questions. But we did end up in the press, and it said, and we had a little article in the Irish Times to say the only known plant to survive the Ice Age. We did end up in an amazing documentary called Secrets of the Irish Landscape, and there's a book. I didn't actually print out my PhD thesis, which I regret, but I do have that book. I have my PhD in a PDF, so I was like, that's fine. I'll take the book. And then hopefully you've got a better understanding of a kind of when the last Ice Age was, where did it go, and how do we know what happened? Now, I'm just going to say, I'm going to finish off this. I wanted to talk about my little squiggly career, and I'm going to leave it at that. But what I do want you to take away is kind of, in the career journey, I'm now at a place in which I'm running an organization, and oh my goodness, do I feel like I'm doing another PhD. Like, so everything I treat as an experiment, and everything I am absolutely using the scientific process. So basically that's just me graduating from my PhD, wondering what to do next. I love teaching, so I became a teacher. I then had babies, and then I realized, actually, it's funny in hindsight, looking back, I loved the analysis so much. I actually pivoted into tech and data. And ultimately, yeah, I won't go into too much about 5-Hour Club because again, that's a whole other thing. But what I will say is, oh my goodness, the transferable skills that I gained along the way, I'm using them on a daily basis now. It's actually incredible. I treat everything like an experiment and everything. So don't underestimate the skills that you pick up when you're studying science, whether that's a degree or a PhD. You don't just learn the knowledge. You may not remember the knowledge in 10 years to come, but you will always be a scientist and you will always have those skills. And I think I was just in a scientific bubble for so long, I took that for granted. Now I'm outside that bubble and I realize it really is like a specific set of skills that you gain that will always stand to you irregardless of what you do. So yeah, I hope that makes sense, trying to cram 4.5 years into 50 minutes. But yeah, if you've got any questions.
Oh, Emma, like I gave you a challenge there and you absolutely exceeded my expectations. I've made so many notes. Like I'm literally, I enjoyed that a lot. I'm going to pass over to the students. Has anyone got any questions?
And I'd like to say there is no such thing as a stupid question. It can literally be on a day-to-day basis, like absolutely anything. I may not be able to answer, but honestly, yeah, any questions whatsoever.
Just while we're waiting, I'll give people some thinking time. Emma, I remember doing gels at uni, and that was like my longest lab. Like, how long timescale was it? Is it similar? We were waiting sort of 8 hours for gels to run. Was it similar for you?
Yeah, that's a really good question, actually. Because no, they weren't that long. Because I do remember, they would actually only be about half an hour to I'd say an hour maximum. But the problem is, you'd set yourself that time, you go for lunch, and then sometimes there have been occasions, I know, you're like, oh no, it's going to run off. It's run off. I've missed it. And that's the other thing. Like, I remember like trying to do so much, like at the same time, I learned the hard way, you have to be systematic. Like, even though it's like, it's actually like relatively simple what you're doing, you have to do it in that way, because you're doing thousands and thousands of, for example, DNA extractions. You have to do it and tick it off, because it's so monotonous, you will just make a mistake, and you want to decrease human error.
Definitely. I remember I did a little teeny tiny bit of research for one of my old lecturers over the summer before I became a teacher. And I remember I like set this gel up, and I was like, right, I've got a few hours now, so I can like nip out, go and get some lunch, come back, like you said. And I set it the wrong way, and it just ran off into the
Yeah, I'm going to be honest. I think yeah, I think that was one of my, because I also, like yourself, I did a little bit of research pre the PhD. Yeah, yeah. And I mean, I think with the trying to make sure that the centrifuge is balanced and all of that, I think that's the other thing with science that you learn, is that you learn more by making mistakes, and you learn more by a negative result is still a result. It's not going to be positive results all the time, and you learn so much from that.
Definitely. And I think by being like a little bit cat-handed sometimes, like you make a mistake, but you only make it once, don't you? Because you're
Yeah.
like, oh, that's so annoying.
Yeah. It's just so true. And I know I said it before, but I absolutely whizzed through like the techniques that I used, and that. So I hope it wasn't too complicated.
No, and we cover some of that in some of our units. So I think this is going to be an amazing resource for future to give them that breadth and depth to be able to talk around like how this is used. One of the units on the legacy. So anyone on the legacy pathway, there's a whole bit on gene technology. This would be a perfect example. You've got to compare two uses of PCR. Like this would be an excellent example, and especially those people studying a sort of more going towards doing an environmental science degree. I know there's a student who's studying with us at the moment who I'm sure is probably working tonight, but is going to be watching this recording and be absolutely buzzing and probably really frustrated he can't ask you a million questions. So I'm definitely going to have to invite you back at some point to address some of those. I love as well the piece about like the variety in your PhD. Like, I don't think, I mean, so some of my other friends who have studied PhD, so one of my friends, Yasmin, looked at the effect of drugs on, you've probably seen the research in the papers in Portsmouth. So she started some of that research of looking at the effects of drugs on shrimp, because they said that the waters in Portsmouth had surprisingly high levels of cocaine present. And they looked at their response to light. Yeah. So I guess her PhD was kind of similar because she'd be out collecting samples. But then she spent a lot of time. Hers was kind of like in the harbour, in the lab, at home, and analysing and writing.
Yeah.
And I guess yours had such diverse, you were using multiple techniques. You were like weaving together like hiking, mapping, the population genetic, like that's huge, isn't it? Is that found in other people's PhDs, or is yours quite unique?
Quite unique, to be honest. Generally, like, I mean, ecology is a little more exciting in one sense, because you get to like do your field work in the summer, and you get to go around and and do whatever that is. Like, so for example, my husband, he's a biology lecturer, and I know for his PhD he was going to forests, but I'm not gonna lie, like I chose my PhD for many reasons, and I'm very grateful. But one of the reasons was because my fourth year thesis, I was in the bioinformatics lab, and I downloaded genomes, and I actually felt like I had the confidence to do the analysis part for this. I did a little bit of research in the lab before my PhD, and it was actually looking at oh my goodness, I'm trying to think back now. It was to do with gene flow and trying to understand about genetic technology and how. Anyway, sorry, this is absolutely on a tangent. It's a very it's a very long-winded way of saying it was unusual to have that combination of the field work, the wet lab work, and in a lot of the labs around me, they were just in the wet lab day and night. But as you can imagine, there's obviously a lot of challenges and a lot of risks and things that can go wrong. But that's why for my particular PhD, I think it was very important early doors, because my PhD supervisor was actually, I think, relatively new scientist. So that's the other thing. If you're choosing a PhD, like, are you going to go with someone who's really well established and it's more like a job, or are you going to choose a big massive adventure and have to have a lot of initiative and try and figure it out? And what I didn't mention was this was in conjunction with the Botanic Garden in Dublin, so I did some work there, and that was great on really cold days. I would just go to the tropical house, and I also wrote a grant to get I got a grant to go to do research in Madrid for 3 weeks to try and understand the technique of AFLPs. So I realized now it's so nice to reflect on it, like how lucky I was to do a
Well,
tier one, to travel and study at the same time.
Well, you make your own luck as well, don't you? And I always say, oh, I've been lucky with my opportunities, but you haven't. You put yourself out there, and actually you looked for what you wanted, and then you were like, there's a grant available. I'm going to go to Madrid for a few weeks. Like, lots of people are like, oh, you're so lucky, but no, like you applied for it, you got it. It had synergy with the work you were doing, and so therefore you were awarded the grant. Like, that process is not an easy one to navigate as well. So
yeah. I mean, you are absolutely right, and ultimately it does come down to problem solving. And I knew that I wouldn't, like, such a massive project. I knew that I couldn't do it by myself, and I would absolutely need all the help that I could get.
Yeah, and those key skills of collaboration. I think back to our Royal Academy days, that was like collaboration in action, but you were so well suited for that. And I can see why Scott and possibly Dominic, I think Scott would have recruited you, didn't he?
That's right, yeah.
Yeah. And I can see why he'd have been like, I bet he heard, like, did you use this as part of your interview? Were you talking about your experiences?
I don't think I even did. No, I don't think I did. But I mean, I'm not gonna lie, like, yeah, because it was project management and collaboration. I think were absolutely key. And I think, like, to my core, I realized that is a fundamental part of like collaboration, for everything. So it was well suited, I think.
And to be fair, like you said, actually, the things that you do, like, I reflect sometimes on stuff that I did ages ago, and I'm like, oh yeah, I did that. I could have talked about that, and I just didn't think about it. But it's the skills that you draw forwards. So actually, I suppose when you were doing the work at the academy, I can't believe that we never spoke over this at lunch break, but then we were probably too excited talking about the collaborations that we were doing in our regions. Like,
yeah. But that's exactly it. And you move on and you forget in a way that you did this large body of work, and you forget how exciting it was at the time, and all of the but one of the reasons why I was thinking about it more recently, and it's so interesting in setting up an organization, so that's 5 Hour Club, genuinely everything is an experiment, and more so than ever, I'm using the skill set that I had from my PhD. So, project management, collaboration, the scientific thinking, like it's incredible.
Yeah, and definitely, I think Jordan and I found the similar sort of thing from setting up Community. This is like a completely different world that we're navigating. When we were like, we'll just set up a charity, but then I was like, well, we'll just have to read about how you do it. We'll have to find the method. And then we couldn't find the methods. Then we invented our own method.
Yeah, exactly. It's just having that initiative and doing the research and thinking on your feet and all of all of these things. That's it. Yeah, I had a feeling you might have found a similar thing, but I really do think it will stand to you having that scientific mindset. I really do think so.
Definitely. And I'm just going to check in because I realize I'm like stealing all of the question time. Does anyone have any questions for Emma? Celestina?
I had a bit of a I had a bit of a silly one. Do you have a favourite organism that you studied the DNA of?
Do I have a favorite organ? No, that's not a silly question at all. So it would have to be, this little hardy Arctic alpine plant. I mean, it has to be, because it's just the incredible story that it has behind it. So for my fourth year, like my undergraduate project, I downloaded the lactobacillus genome, and it was really interesting. And but I think this just had such a ridiculously good story. Now, what I didn't mention was there were 6 other plant species that we also sampled around Europe. And originally, this is the thing, like things change. Originally, it was going to be 8 different plant species, and we're going to do analysis, and then we realized, well, we have a lot of data, we can just do 2, and then we put them in the minus 80 degrees freezer, so there can be research for years to come. But there was one called Monartia recurva, and oh my goodness, did we get lost for the like in the middle of Scotland, and it was on this, and this was a really rare plant, so we had to get like permission to sample it, and it was like raining and there was a cliff. But no, I think the winner is Arenaria ciliata. Definitely. Yeah, it's got a great story.
I love that. I used to have a sign in my lab when I taught in school, and it said, the people who ask silly questions become scientists.
Yes. Oh, that is such a great question. That's another, not like the being inquisitive, like that, honestly. Yeah. It's definitely, again, it's just something I took for granted that everyone is, but not necessarily.
No, definitely. And I do think, those kids that ask questions, I mean, sometimes the ones in my class, I'll be like, right, can we just stay focused on the thing that I need to teach you, because I'll go off on a tangent. But at the same time, I'll be like, your curiosity is so beautiful. Do not lose that. That is such a skill. Yeah. And actually, you do have to almost practice it, don't you? I can find myself like, when I became too shoehorned into an exam-based system, I was very much like, I probably lost my own curiosity a little bit. And then as I've stepped away from the classroom and done other things, I'm like, oh, I wonder if, and like it really, it makes a difference in the way that you behave in every aspect of your life.
It really, really does. And what I didn't go into too detail was that I'm also a data consultant and a business analyst. And, part of that role is interviewing people and understanding and really, again, that curiosity and to dig deeper, and all of these skills are so needed, and especially in this world of AI and etc., these soft skills that you develop, interestingly enough, as a scientist, that that are potentially innately in you, but are also shaped by doing something like a degree and a PhD or a master's, they're always going to stand to you. But I don't think I could have said that 5 years ago. But, yeah.
It sometimes takes a little bit of time out and some reflection as well to really understand and appreciate. Like I talk a lot on my courses about that metacognition, like understanding how you learn, and I think actually it's really hard. And actually, I don't think I truly understood what made me better at teaching until quite a few years in. I think after I'd finished my master's in education, I was like, oh, like I think I'm beginning to get it now. And then it was only a few years later that I was like, no, I know what it is. I know what the special sauce is. I know what works and why it works. And I think, and then that works for your own skills, doesn't it, as well?
Absolutely. Like yeah 100%.
Celestina, I don't know if I'm saying that right, but have you got a question? I can see you're unmuted.
No, not now.
Not now.
It's a lot to take in right now.
Well, one of the beautiful things of our courses are as well, of course, this is recorded. And I think one of the really nice things that you can do, so we've got a couple of other guest lectures, so you can go and watch those as well. But actually, in a month or so's time, or maybe a couple of weeks' time, re-watch this. And I think you'll probably find that you pick up different elements and different things from it. But this is such an amazing resource, Emma. Thank you so much for coming on tonight.
Thank you so much. My God, so much to take in.
Yeah.
Awesome. Thank you.
Celestina, what course route are you on? Oh, if you unmute, Celestina, what course route are you on?
Nursing.
Nursing. Oh, amazing. Do you know what though? Lois, who joined us, who's in her second year of nursing now, she came to every lecture. So she came to everything. So I was delivering, all of the stuff that people were going on to do environmental science with. She came to everything and she was like, I just have a thirst for knowledge now. I love it.
Yeah. I want to do mental health.
Amazing. And actually there's a huge piece around the environment and the impact of being around in nature and appreciation for nature linked with, improving health and well-being, isn't there?
Yeah, I actually came to the course because I wanted to do like a postgraduate diploma, and they asked for a nursing, if I have
Excellent.
any experience with any course in nursing. That's why I came to do this course.
Amazing.
Because I shouldn't have done a master's in business. I wasted all that money. I should have just done nursing. Wasted all my money and time doing masters in business.
It's never wasted. You'll
My
draw
heart,
across
yeah, I
skills.
know. My heart is really in like mental health because so many people right now. It's the other day I was going to Scotland. We at the Euston station. Someone jumped in front of the train, and we were there for hours. And I said to myself, "Oh my God, something must have triggered him," you know. And then my daughter's friend, the other day, she just like she had a total meltdown for 3 months. I was like, "Oh my God." So I said to my daughter, "You know what? I'm going to do mental health nursing." She said, "Mom, you've been saying this for many years. Go on then."
I
So
love.
yeah, so when I tried to apply for postgraduate diploma, at least start somewhere, they said, "Oh, I needed a course in, no matter how basic it is, first." So I started searching online and I found this one, which I'm really happy. I'm really looking forward to actually doing it to the highest level I can, because I really want to. I'm a people person.
Oh,
I used to walk my
Celestina,
country.
I will get Lois back to come and talk to you. 'Cause she's got a very similar story and she spent her
Okay.
life doing all of the other things that worked around her
Yeah.
family. And then she said, I want to be a nurse. And
Yeah,
that
I
is,
even did
so,
this master's in psychology, and I thought, nah, this is not me. I
I
actually
love
want
it.
to do nursing because I have a cousin that is my younger sister is a nurse in London. She does
Amazing.
the ICU one. Actually, it's just like mine is. I want to do the mental health section of it because that's
Amazing.
where I think we really need to help people. It's
Massively.
People are going through so much right now
Oh,
that I just
well,
want to. I
that
just
gives
want.
me, oh, sorry.
I just want to be in that field to be able to like help people, listen to them, and see how we can actually help them to overcome all the. Because years ago, I worked at the job center in Croydon,
Oh,
and I was put in charge of the homeless people.
yeah.
So I suggested to the manager then, I said, why don't we actually have a day where we sit down with these people and find out exactly how they became, like, got
It's
to
like
where
find
they
a,
are?
yeah.
Because when they come in, you can
Yeah.
tell they like want to talk to you. They just want somebody to listen. I
Love
said to
that.
him, I said, if we talk to them, find out the situation, help them get
Yeah.
back their confidence and help them back into work, because a lot of them were cut off from their families. They couldn't see their children. So the manager agreed, and then there was a week of blitz where I had to sit down with so many of them. In At the end of the week, we got a lot of them back into homes, so they were
That's so
able to see their
lovely.
families, their children, and then they picked
Yeah,
up their confidence, and we helped them with
that's
their
beautiful.
CVs and help them back into work, because
Yeah.
you don't just become homeless because
Celestina, I'm just
yeah.
going to stop you there for a moment
Okay.
because I'm going to wrap up. I'm going to stay
Okay.
online. So I know you've just recently joined, so I will stay online to chat to you in a moment. I'm
Okay.
just going to let Emma go because I appreciate and imagine that possibly bedtime going
Yeah.
okay in your house?
Yeah, I just heard some footsteps downstairs, and then I'm sorry, my alarm went off because the garden waste took place.
No, No. Emma, thank you so much. It has been incredible hearing you speak this evening, and like even as your friend, like I'm seeing I didn't know about all your research, and I've just enjoyed it so much. So I'm so grateful. Thank you so much.
Oh, you're so welcome. Anytime. And yeah, I hope it's difficult to get the balance between the detail and obviously being an ex-teacher. And I'm like, okay, I'm not explaining gel electrophoresis.
Yeah.
But be very aware you've got a whole hour lesson that you could do on it. So no, I hope
Oh my goodness. No, you absolutely nailed it. You pitched it perfect.
Oh, well, thank you very much.
Thank you so much. My God. Thank you.
No, you're very welcome. And I will. No, you're very